Well, thank you for the intro, and thank you all for coming. And so, let's see, Ben already kind of covered what I'm going to be talking about, at least with the title. And so just a little bit of disclosure stuff. So Genome Oncology is sponsoring this event. So we all got lunch because of Genome Oncology. I'm not being paid to give this talk at all. Genome Oncology has never paid me anything. And I actually pay them to use their stuff. So. And then also the work that I'm talking about and the presentation itself was not vetted at all by Genome Ecology. So, although it's not like a slander campaign, so you guys don't have to worry about it. But in general, you know, this is all very independent work. So, you know, a lot of what we've done in our institution, Children's National, is driven by the explosion of patients that we've seen in our division of genetics and metabolism. And so this division, we were seeing about 1,800 patients a year in 2009, and then through a very rapid hiring process and recruiting process of board-certified geneticists and genetic counselors, we've managed to turn ourselves into the focal point of genetic testing, of genetics and seeing patients with genetic diseases at Children's National. So the driving force between our testing lab has really been the clinical front. And so as this division of genetics and metabolism who sees the patients and orders genetic testing, you know, as more patients have come in, we've seen a very big explosion of genetic testing at Children's National. And so, you know, our role in the institution, like many other diagnostic labs, is to bring this business inside. So whatever testing we're not doing is being sent out to other reference labs or other institutions around the country. And so when those tests are sent out, the way it works is our hospital gets institutionally billed by each reference lab, and then we, in turn, bill the patient and the patient's insurance company. And so if you look at all of the sequencing tests that were ordered in a one-year time frame, there were around 800 different sequencing tests. And so you know, this is good because it's a solid volume of testing. And when we're talking about bringing testing in-house and we're talking about expanding our services, you know, we really want to be in a situation where we have enough business to support the growth of our program. And so when you look at the total number of tests, it's great. 800 tests is a very solid volume for a small institutional lab like ours. However, when you look at the spread, this is not 800 of one test. What we're seeing is, if you look at it, many, many different tests are being ordered every single year. And so this graph right here, what it's showing is that the most common test, which is at the top there, was ordered 58 times in 1 year. There were 257 tests that were ordered only one time. And so if you're thinking about developing and validating tests, if a test is only ordered one time, you're never going to develop and validate a test for that. It just doesn't make sense to develop a test when you're only going to be getting one sample a year. And so what we wanted to do was, you know, we really needed to figure out a way to address this because, once again, our job is to bring this testing in-house. But the way it looked right here is there's something like 300 or 400 different tests that are being ordered, and no tests are being ordered high enough volumes to actually support growth and insourcing. So, you know, as I said, the problem is that to bring this testing in-house, we would need to develop and validate hundreds of different tests. These are single gene tests. These are multi-gene panels. There's whole exomes in here, although our institution in general tends to order whole exomes as more of a test of last resort. We tend to focus on panels mostly. And then there's also the need to have a huge amount of R&D personnel, space, and capital. And then even with this, it would be immense.
Plug it in again.
Sure. Are we seeing this? All right. Sorry. This is, you know, just to give a little bit of background on technical issues. So if these are the only technical issues, it'll be surprising because we're going to be rotating between 3 different applications. And then there's also, you know, the actual presentation itself that we're seeing right now. But so you know, basically it's infeasible to take the old system of developing lots of different tests to meet the needs of our patients, because to look at this, we would never be able to develop hundreds of different tests, and we would never be able to get enough volume from developing any one test to actually support bringing this testing in-house. And so what we needed to do was figure out a solution to this. And so our solution is to use high throughput sequencing and we sequence one large set of a comprehensive set of genes for all patients. Okay, so every patient, no matter what sequencing test they're getting done, we're always going to be doing the same library prep and the same bench work. And what that is, in our case, is a 4,800 gene panel. And so the idea is to create a comprehensive enough panel that any given clinical situation would apply. And then we take a personalized approach, and we're analyzing only variants in genes that have been requested by the physician in that patient that's clinically relevant to that patient. So if we are working a kit up for cystic fibrosis, we will sequence 4,800 genes, and then we only look at mutations in CFTR. And if we're working a kit up for epilepsy, we're going to sequence 4,800 genes, but we're only going to look at maybe the 80 most common epilepsy genes. And so, you know, this same model applies for every single different case that's being worked up in this whole entire spread. And, you know, it's a very personalized approach where we're only focusing on clinically relevant genes. And the benefit of that is that the more genes that you are looking at, the more variants of uncertain significance that you are going to end up with in a report. And so by focusing the genes that we're looking at to the patient and not trying to come up with the most comprehensive list of genes that we could possibly come up with, we end up with less variants of uncertain significance. And so, you know, the benefits to us are that we're able to develop and validate a single test to address the whole scope of all those sendouts. And so, as I said, we're running a 4,800 gene panel, and this is at the time that it was made, it was all the genes that were associated with a clinical phenotype. This was developed from sources like OMIM, HGMD, GTR, every different clinical lab, and kind of just throwing everything together. It's not a whole exome, okay? So we're not sequencing 25,000 genes. There's no whole exome backbone on this. It's literally just clinically relevant genes. It's a relatively easy workflow to follow, so it's based on Illumina's Nextera rapid capture technology. It's amenable to automation. And if you look at these 4,800 genes, you know, almost all of those sendouts, which were shown in that graph, are covered in this one test. And so you know, this is what we ended up doing in our lab. And so, you know, when we had this idea, we wanted to have kind of, you know, I had this process flow in my mind of how I wanted it to work out. And you know, I'm going to kind of walk you guys through this, and as I hit each point, I'm going to show how we've been using Genome Oncology software to kind of meet all these. At the time that we had this whole flow chart set up, you know, as Ben mentioned, Genome Ecology was not doing any inherited disease testing. It was all somatic testing. But we thought that there was enough value to explore whether this could be applied to inherited disease situation. So, you know, the process starts when a physician has a patient in need of genetic testing. And then, you know, if the physician has a patient in need of genetic testing, they have the ability to order one of 29 configured tests that we built in our system. And so what we did was we took the 29 most common orderables from that whole big spread that was in that graph, and we created on the top portion, so the most common ones, we created individual orderables for each one. Now, as I said, that whole graph was showing something like 300 or 400 different tests. And so there's going to be a lot of scenarios that are not covered under the 29 most common tests. And so for those, we wanted to, sorry, this is just a little bit of our rec sheet. So these are the 29 that we configured. So you could see, you know, some of these are single gene tests like CFTR sequencing. There's a bunch of cardiomyopathy testing, a bunch of epilepsy testing, hearing loss, osteogenesis imperfecta, connexin panel. And so, you know, the list goes on, but it's really going to be your most common testing that you would expect to see from a pediatric hospital. And then for the situations where the configured tests were not able to, where the patient didn't fit the mold of one of these 29 configured tests, we created what we call personalized sequencing panels. And these personalized sequencing panels are kind of like a wild card. So when somebody orders a personalized sequencing panel, they can provide us with a list of genes that they want us to analyze and interpret variants in, or they can give us a phenotype and we can establish the list of genes for them. And then when we actually, so then the follow-up is that we would end up running the sample and looking at whatever genes are established in the front end. So this is a lot different than a whole exome approach. So when you think of a whole exome approach, you start with, you know, all of your variants being in some kind of like master list, and then you parse down and narrow down that list as much as you possibly can using all these different features through filtering to the point that you end up with like the handful of most clinically relevant genes or clinically relevant variants. In our case, we're doing this on the front end. We're establishing which genes we're going to be looking at, and this is before the test is even run. And the reason that we wanted to set it up before the test is even run, sorry, this is just what it looks like in the rec sheet. So you can see there's one option for gene list, and there's one option when it's a personalized sequencing by phenotype. There's a minimal premium that's basically tacked on to the difference between gene list and phenotype, and there's just to really account for how much effort it's taking on our end to develop these panels. But you know, so the reason that we do this beforehand, because we want, before we run the test, to be able to say to the physician, you know, is the gene that you've requested included in this 4,800 gene panel? So as I said, at the time, it was very comprehensive. At this point, just because of the speed of advancement and the number of new genes that are being associated with different clinical phenotypes as time goes on, you know, it's a snapshot in time when the panel was created. And so there's going to be the occasional gene that's not covered on our panel, and we want to be able to tell the physician that before we run the test. Because if it's a gene that they're really, really, you know, gung-ho on getting sequencing on that gene and it's not on our panel, then we don't want to go through the whole process, write the report, and then have them realize at that point that it's not covered on our panel. It would have been kind of bad for us. It would have been bad for the patient. And then the other question that we ask is this gene covered well? And what we consider as being covered well is, what we're asking is, are there any spots in the requested genes, any amplicons that have the potential to be low covered spots? And what we're basing this on is basically all of our validation data, where we've run hundreds of samples. We pooled all that data together, and we have a good idea of areas of our medical exome that are going to have low covered spots. There's not very many of them, so 95% of our 4,800 genes are covered at greater than 20x, but there is the occasional amplicon. Usually it happens to be an exon 1, as we all know. They're harder to do NGS on. You know, but we want to be able to, before we run the test, tell the physician, yes, this gene is on our panel and it is going to be covered well. And sometimes when we tell them that it's not, that one of the many genes that they're requesting are not on our panel, they will still proceed with our test, and sometimes they won't. And in the cases that they won't, then they're perfectly capable of ordering the test as a send-out. So we're not saying everybody has to do our test. We're just saying, when equivalent, order our test. The other option, when you look at the by phenotype option, is, you know, what genes fit with this phenotype. And I'll show a little bit of how this works in a second, but we use sources like Human Phenotype Ontology Database, OMIM, PubMed. We look at other labs' panels and see what they're offering. We also use regions of homozygosity that are generated from chromosomal microarray that we're running on every patient. Sorry, not every patient, but most of our patients. Typical first-tier workup in our institution is to order a chromosomal microarray when there's not a single genetic suspected disorder. But anytime that there's intellectual disabilities, autism, epilepsy, dysmorphic features, arrays are always ordered. And then as a follow-up, oftentimes sequencing is ordered. And so what we want to do was be able to take our array data and feed it into our gene panel builder and decide, you know, which areas of homozygosity in that patient are going to be looked at, which genes in the areas of homozygosity are going to be looked at, because those are going to be hotspots for recessive conditions. And then we wanted to have the ability to generate like a pre-test report. So to say to the physician, you know, this is how we generated this panel. This is what's on our TrueSight 1, which is the library prep that we're using. This is what's not on it. These are areas that have potential to be weakly covered, and this is how we generated it. So, just looking at the Genome Oncology software, I'm going to kind of go into here now. So this software, this is the Genome Ecology software. It's set up in these modules, and so the first place that we start is this ordering portal. Sorry, and we're actually going into the application now. So okay, and so this is what it looks like. So what we're doing is we upload in to the Genome Ecology software a work list. So these are all the orders that have been placed on all the patients. Okay, so this is obviously, for the demo purposes, we loaded in non-real patients. But what you see here is that this first line here is an order for one of the personalized sequencing panels by gene list. Now, this second one is one of our configured tests, and in this case, it's Noonan syndrome panel, which is a 13-gene panel. If you look at the personalized sequencing panel, you have this option to select the test, and this is where we're really building the panel. But with our configured tests, so the Noonan syndrome, that's not actually an option because we're only going to look at those 13 genes because that's what the physician ordered. So when we look at a personalized sequencing panel, this is how we actually end up building the panel. So, there's different ways of doing this. The first way is that we can start with any of our tests. So we could start with any of the 29 tests and add or delete genes from there. So each of these are a different test order code, and we could basically build the panel that way. We also have the option to add in different by phenotypes. So for example, if we type in lactic acidosis, and so this is using human phenotype ontology. And we see here lactic acidosis has 106 genes. We then add these genes in, and basically these are the genes that we're going to be looking at in this patient. And so what you see here, and this may be a little hard to see in the background, but what you see is that each gene is listed here, and these all link out to I think it's set up to link out to genenames.org, which is kind of like a clearinghouse for NCBI, Ensemble, and a whole bunch of other places that you could see where it talks about the different genes. It talks about it shows which phenotype. So we search for lactic acidosis, and these genes were added for that purpose. You could see that CA5A is not in TrueSight 1, so it's not going to be covered in our library prep, and we know that right away. So if you look down here, 106 genes are involved in lactic acidosis, but only 93 of them are on our library prep. And so, as I said, we want to be able to tell the physician this before we get started. And I'm actually going to show a case where we talked about lactic acidosis. But so just moving on, the other option of building, I'm just going to clear out this gene list, is to pick an actual syndrome or a phenotype. So this is OMIM database. So this is basically, you could search for any single, different OMIM syndrome. So I'm just going to pick kind of my favorite rare disease. May have spelled propionic acidemia wrong. There we go. And so here are the two genes that are involved in propionic acidemia. So this is basically just hitting up the OMIM database. And actually, if you go and you click on this, it'll actually link out to OMIM itself and show you, this is the disease and these are the genes that are involved. And they're both covered on TrueSight 1, so there's no red. And then we have the ability to just type in any single genes that we want. So we could put in CFTR and APC and whatever, RET, and then we can add these genes on. And so this is basically what we do is we're going through and we're generating a panel of genes that we're going to be looking at in the patient. So this is all before we've run any single test. And that's an important thing to realize is that what we're trying to do at this point is just to establish which genes we should be looking at. So the last way of getting data in, which is pretty cool, and this has been a lot of, there's been a lot of actual interest in this in our institution because of the number of chromosomal microarrays that are being run on every patient before sequencing, is that we have the ability to actually upload in some array data. And so I'm going to do that right here. This takes a second because it's running on a virtual machine here that's just on my laptop, so it's not nearly as fast as the way that we usually run it. Hit the import button again. All right, try this one more time, and then we'll give up on this. Okay. Well, so anyway, what happens is you're able to actually pull it in and look for areas of homozygosity and use that as a primary filter. And then what you could do is parse down and look for different genes in these regions of interest, which in our case are mostly from array data. And so, what this allows us to do is to generate, depending on the patient, we can generate a unique test for every single patient that we're looking at. And then, what we have is the ability, and I'm just going to go back to lactic acidosis. So we have the ability to generate a pretest report. And so what this pretest report is it would have all the patient information, and then it'll have all the genes and why we've added them in. And it'll say, so this is basically using just the human phenotype ontology for lactic acidosis. But if we had some that were from OMIM, or some that were from a pre-existing panel, or if there was a region of interest filter as well, that would all be included on here. And so we could give this to the physician and say, this is the panel that we think that you should be running on, that we think you should be ordering. And these are the genes that if you approve, we'll actually look at in this patient. So that's the ordering portal, and once we actually save the order, it's then tied to that patient. So what happens is anytime that the order is saved, then we'll go and actually run the test. Let me show that. So then, it's in gray here, and it's not expected for you guys to go into and kind of check this all out. But what we're doing is, then we would do the DNA extraction. Then we do our library prep, our sequencing, our alignment and variant calling is all happening in the cloud with data storage. And then what we do is we pull all that data, or not all the data, but some of the data into Genome Oncology for what we call a QC review. And so I'm just going to show the QC module of Genome Ecology. Okay, so this is a run that we looked at, and what happens when we're reviewing the run is that we're actually looking at kind of two different levels of data. So we have run level QC metrics that everything has to pass, and so you see that here. And these are things that are for the whole run, like clusters passing filter, percent Q30, what the passing filter cluster density was. And anytime that one of these don't pass, it ends up showing up in red, and you can see that down here. And whenever something doesn't pass, it requires a note to actually get through the QC so that we could see variance in the report. So the benefit of this is that there's a built-in QC check, and when somebody overrides a QC failure or a QC flag, then all that is tracked. And so if I am reviewing the QC, and I see, this run looked good, but one of these metrics are right on the edge, and so it's causing a flag, I could say something like, okay to pass, and then that would be an option, and it would show up. And it would track the fact that Sean Hofherr went in at this time on this day and approved this run. And then, in addition to these run metrics, we have actual sample metrics. And so these are more based on the whole entire sample and not on the run level. So here we're looking at how many variants were called. What is the mean region coverage depth? What's the read enrichment? What's the percent Q30 for the sample? And we have all these different things. And once again, if something fails for whatever reason, we have to go in and put in a comment. So this is actually all the samples in this run. Some of them had flags, and these flags were overridden during the review process, and then a note was written in place. So what this allows us to do is actually to have tracking of the QC metrics for every single run that we are doing, which is very important. It also lets us go back to the sample and say, okay, something weird happened in this sample. Was there anything in the QC that should have told us that something was weird in there? And it's really good for traceability. And in a clinical lab, you really need to have this kind of auditability, and you need to be able to track your QC for every single sample. Okay, sorry, I'm getting a little warning here. Okay. All right, so what happens, so once the test is run, and QC is passed, at that point, the actual panel that we've created in the ordering portal is applied onto the sample. So until this point, everything, so when we were looking at those QC metrics, we were looking at QC metrics for all 4,800 genes. Once we're at this point, at this level of variant analysis and interpretation, we will only be looking at variants in genes that have been established in that ordering portal that I had shown. And then also the same thing with the generation of the clinical report. So we cannot look at mutations or variants in genes that have not been ordered before we ran the test. And we cannot put variants into a report that have not been ordered. So this is really important because incidental findings are a huge issue. And if you're looking at every single variant, even if you're only looking at one gene, but yet the first view that the director has is all the variants, and you see something that's clinically actionable, then the responsibility of the lab director is to follow up on it. And so because everything is completely masked, it really creates this distancing between incidental findings where there's no way for me to even uncover the incidental findings because they haven't been ordered in the patient. So I'm just going to show, and this is kind of the Workbench is really the meat of the Genome Oncology software. And this is where we're actually going through all the variants that we're looking at in any patient. Okay, so these are 4 different tests that we had run. So some of these are configured panels, and some of them are going to be the personalized sequencing panel. I'm just going to go through one of them to kind of show what it looks like. But from here, it has all the patient information. And so what happens is our data files, when they're living on the cloud, all have a unique identifier that's generated from our laboratory information system. And so the data for every patient is labeled with a barcode. And then what happens is that order that we've uploaded into Genome Ecology and the ordering portal, it ties together with the data through that barcode. And so they marry inside of our firewall. And so there's no actual PHI that's associated with any of our data until it comes back into our system and it actually goes into the Genome Ecology software. So this is a sample, and so what it looks like, and this is kind of the director view, and so this is where I spend most of my time. And the first tab that I see, because I'm usually not the person that's going in and reviewing the QC, so usually that's one of our staff scientists that's going in and saying, okay, this looks up to specs, and this is the reason that I'm going to accept a run. So all of that is not usually happening by me, and so what I end up seeing is I'm going through all the variants, but the first thing I want to see, which is this first tab, is how was the QC for this sample? And so up here we're seeing how the run QC was for this sample. And then down here, we're seeing how this sample's QC was as well. And then, there's some cool things built into this where, I can look and I could say, okay, so this is every single amplicon that's been ordered on this patient. And if you look here, there's one amplicon that has only a coverage of 5.4. And so it automatically gets flagged as a failure in Genome Ecology. It still shows up as if it's a variant, if there's a variant called in there. But it's there so that I know there's a poorly covered area. And so, that just it's something. And then this goes into the report as well. So any poorly covered areas of genes that have been requested based on the actual data on that patient will go into the report. So remember, in the ordering portal, everything was getting based off of our validation data. So it was a prediction. So we didn't know how that panel would be covered in that patient. All we could say was, based on these hundreds of patients, these are any kind of potential weak spots. But here we're actually looking at that patient's data and we see, okay, there's one amplicon that has poor coverage. So then, as you can see, it's all tab-based. And so when we're going through, there's different tabs. So in this case, pathogenic, likely pathogenic, uncertain, intronic synonymous, likely benign, common, benign. Those are the main ones that our variants are getting bucketed into. And so we've set up rules that dictates how some automatic sorting is happening. And the whole purpose of this is to really let us leverage all the patients that we've previously run and all the variants we've previously seen, but also how can we leverage things like frequency databases that a very frequent variant that's non-reference, is it really significant if it's at 50% in the population? And the answer is no, at least not for the rare diseases that we're looking at. And so we've set up these rules to basically determine where our variants are going. And so for a variant in our hands to go into the pathogenic tab, it needs to have previously been called pathogenic by us. And so for each end of the spectrum, for benign and pathogenic, the only way that a variant could get into either one of those tabs is if we've previously seen that exact variant in a different patient and we previously curated it and categorized it as pathogenic or benign. So we didn't want to have any kind of automatic filtering going to those two ends of the spectrum. And so pathogenic is really reserved for mutations that we've called pathogenic, and benign is really reserved for alterations that we've determined are benign. Likely pathogenic is usually filled with variants that's getting cataloged from, in our case, it's based off of dbSNP and 1,000 genomes. And so if another laboratory has seen a variant and it's been logged into dbSNP or 1,000 genomes as pathogenic or likely pathogenic, it will fall into this likely pathogenic area. Now, that doesn't mean that we don't have to review it. So we still go in and review the variant, and then we categorize the variant. And however we categorize the variant, the next time that same variant is found, it will automatically go to that place. But the first time, it's going to go into the likely pathogenic. And it's just a way to kind of automatically sort to kind of cut down on our overall thinking process. Uncertain is any variant that's classified as uncertain, or if there's no classification at all. And so it's not based on rules like effect of the alteration. So for example, this first alteration right here. So I'm just going to walk you guys through it to kind of orient you all. So the gene is COLQ, so COLQ. The position of the variant is basically the genomic coordinates. It then has the C dot, so this is all HGVS nomenclature. It has the protein effect, so there's the alteration causes an arginine to go to a stop at position 227 in the protein coding region. And then the mutation type, so it's a nonsense mutation. And then this count area, so the count is how many times has my lab seen that variant. And if we've seen this variant before, then this would actually be blue, and it would be a hyperlink. And when I click on it, it would let me pull up every patient that I've reported with that variant. Because this is a demo build of our Genome Oncology system that's living as a virtual machine on here, it doesn't have our database. But if I'd previously seen this variant, it would tell me how many times I've seen it and how many times I actually sent it off for Sanger confirmation. Then kind of looking at the effect, it's a G to an A. And then if you look at significance, so it's been called pathogenic by me. So I went in and curated this as pathogenic. Since this is the first case that I've seen it at, it still is living in the uncertain significance bucket, but the next time that it's seen, this variant will be living in this pathogenic area. Then it says whether there's a categorization in dbSNP. It says, what's the frequency in 1,000 genomes, EVS, and exact. And then what we have is a spot that we could go in and interpret the variant. So if I decide that I'm going to potentially be interpreting this variant, I would click on this View Details and Edit. And so this is actually, so this is data from an actual case. And so in this case, I called it as pathogenic. There's a section for internal notes and a section for variant notes. In this case, we just copied and pasted the same information because we were just filling up the demo instance. But this is the actual interpretation that we used on this patient. So this is a free text field where we've said, this patient is homozygous for this nonsense substitution in this gene, and then we talk about what the clinical significance is. It says what the pipeline version was. And so the idea is that the next time that this variant shows up, this information will still be there. And if all of a sudden I realize that maybe I shouldn't have called this pathogenic in this patient, and I see it in patient 2, and I want to edit this, I can actually go in and edit this interpretation and write something totally different. And when I save this, it timestamps, and it stamps that I'm the person that did it, that changed it. I could also choose to only change it for this case. So for example, if you see a variant that maybe you want to build your report where a variant has more significance because there's a second pathogenic variant in the same gene and it's an autosomal recessive condition, you would build the interpretation slightly different for that patient than a patient where you're just seeing a single heterozygous change in the same gene. And so I may say use only in this case, and then it will literally only use that in this case, and the overriding interpretation that I put will be the one that defaults in there. But so the idea is that the next time that this variant shows up, it will take the same interpretation that I previously put in, but it's totally editable. I could also edit the way that I'm classifying it. And then you could add in PubMed references as well. And and these show up into the report. Just showing a little bit more about how we determine significance. So this is using a software called Alamute from Interactive Biosoftware. And so coming from Mayo Clinic where I was, we were very dependent on this software. So this is like our life for determining significance. It's excellent software, so it all works in a dashboard format. This is pulling up the information on this specific variant. So as I said, it's an arginine to a stop in this gene. It gives you the frequency for exact. There's no frequency available for ESP. If there was an HGMD, so that's Human Genetic Mutation Database entry, then it would show up here as well. It has all the variant prediction program, so align, SIF, mutation taster, polyphen. In this case, this is a stop codon, or it's a stop mutation, a nonsense mutation, so there's no pre-populated information. It only comes, you only get prediction information when it's an actual missense mutation. And then you can look at effects on splicing as well. And so in this case, this is comparing reference to alt. And so, the benefits of having this is that we're able to use software that we've become very used to. And we use this along with the Genome Ecology software. And so when we're generating the interpretive comment for this particular variant, we're usually navigating back and forth between all these different sources. So whether they be PubMed, OMIM, pulling stuff in directly from Alamute, looking at previously reported cases, all of that is coming together to generate our interpretive comment. Let's see. Okay, and then some of the benefits of Genome Ecology software is really that it has all of the read data, so it's got all the depth, the quality, the Q-score. it also pulls in the BAM file. So this is very different than a lot of other software that you're using to generate an interpretive report. So there's quite a lot of software out there, and almost every single one that we've ever tested and seen do not have the ability to pull in the BAM file. It's a pretty rare occurrence. And so the benefits of Genome Oncology software is that it's pulling in the BAM file. It has a BAM viewer that's embedded into the software. And we can look at the pileup for any single variant that's being called in the VCF. And so what you see here is that this is a homozygous change. So it's a G to an A. Anywhere that you see a comma, it's the reference. And so this spot right here is all alt. And so, this happens to be a very highly covered area. So there's 303 reads for this particular base, and 295 of them were variant. And so this is just really useful information because we don't need to open up a second piece of software like IGV, load it with the VCF file, load it with the BAM file, and then look for this specific variant. This is really, really appealing. The other kind of interesting aspect of this software, of Genome Ecology software, is that when we see a variant that's going to go into our report, we always want to Sanger confirm the variant. And so to keep track of Sanger confirmations, it's actually embedded into the software. And so when we see a variant that we determine is going to go into the report, or we think it needs to be Sanger confirmed, we then would select confirm. And so as you see, this particular variant right here, which was the stop that got introduced into this called Q gene, has a confirmation that's pending right here. And I'll show you in a second where that actually comes into play. But the cool thing about this is that, as I said, we get variants that are getting bucketed into these tabs. Common variants are getting bucketed in by frequency rules that we put in. Benign are getting put in for the reasons that I said if we previously classified it as benign. There's this tab that says patient, and this is where we put in our recommendations. And so in every report, there's recommendations. And so in this case, it's just your kind of standard boilerplate clinical correlations required or recommended. Genetic counseling is also recommended. But the cool thing is that while we're doing all this, the report is actually getting generated. So this is our report. So this is so this is a report. This would have all the patient's information. In this case, it's a demo, so it says test 4. It has the ordering physician, when it was ordered. It has clinical indication, which we enter in the ordering portal. It has the genes that have been analyzed, and this is also set up in that ordering portal before we run the test. In this case, as I said, there's one result, the one that we're actually reporting on, that's pending Sanger confirmation to come in. And so because of that, this report is showing up as a preliminary report, so it's not letting us actually release it. It is saying here that the result is pending confirmation, and it actually does not show that interpretation that we put in. So, I showed you where we could edit in the interpretation, and that's not showing up here either. So I'm just going to quickly show you what it looks like where the Sanger confirmation information is coming in and why that sample is getting hung up. Okay, so this is a kind of a built-in laboratory information system. It's very, very basic, so don't think of this like your massive laboratory information system that's running your clinical lab. That's not the whole point of this. The point of this is to keep track of things like confirmations. So in this case, this is the test that was ordered and the sample and kind of the information. This was when the Sanger confirmation was requested. This is the actual chromosomal spot, genomic coordinate that we're Sanger confirming. And then what happens is when you want to when you're looking at your Sanger data, so that is not built into Genome Oncology. So what we're doing is we're physically going in and opening up a piece of software to look at the Sanger confirmations. And then what we do is we go into here and we say, it has been detected. And then we put a note like, heterozygous, homozygous, detected by, whoever did it. And then we click submit. And so now that that has been submitted, I'm going to go back to the Workbench. And I'm going to pull back up the same case. And I'm going to go to this variant. And now you see it says confirmation result detected. Okay, so before it said pending. And then likewise, now when I click on this report tab, sorry, everything's a little slower here. So just a little bit of kind of technical information. So we run this software, we run all the Genome Oncology software on a Mac Pro. So our whole entire process is happening on a Mac Pro. And it's, you know, if anybody has seen any of the newer Mac Pros, so it's one of the little cylinder ones, so it doesn't take a lot of space. And that's where everything is being run. So right now, this instance actually was running on an external hard drive. Okay, so it doesn't need to have that kind of processing power, but to allow multiple people to log in at the same time, to allow the database to be kind of going and expanding and let you go from one spot to the other quickly and not lag up. That's why we have everything running on the Mac Pro at home in our lab. So here now what you see is that this is our report. It no longer says preliminary because all the Sanger confirmations are in. It has the result, which is the variant. It has the interpretation. So this is what I went in and kind of had the ability to edit. It has the recommendations that we talked about. And then there's canned language for our methods, our database, our limitations. And then if you look here, I showed that one amplicon that had low coverage, and it's actually showing up here. So this is the amplicon. So amplicons with coverage below 10x, that's what we kind of felt should go into the report if it's less than 10x, and there's only one amplicon that's there. And then your standard disclaimer. We always put a results summary in, and this is because physicians don't actually read clinical reports. So if anybody's ever spoken to ordering physicians, you know, you could put together a 20-page clinical report, and they will read the first like 3 lines of the report. And so we make our first 3 lines the most important lines. And so this results summary box right here on the left that says positive analysis, testing identified a homozygous mutation in this gene. This finding is a known pathogenic mutation is consistent with this disease. That will show up at the very top. And so once I finalize it, oh, sorry. So this is another cool feature of this software is that every single variant needs to be looked at. And every single variant not only needs to be looked at by the genetic counselor who's kind of doing the first pass, it also needs to be looked at by me. And I need to say that I've agreed with the genetic counselor or the genome analyst or whoever it is in your lab. And so in this case, there was still one that was pending. And so I'm just checking it off, so saying that I agree with it. So it doesn't mean that you have to go line by line and agree with every single thing. I usually do it in batches with the common variants one because the genetic counselor has already gone through them. Just making sure that I've actually gone through and accepted all of the changes. And now when I click on finalize, now you can see the results summary is actually in there. So this is the physical report right here. And it's just getting built as we go. That's kind of the cool thing about the software is it's not that we're doing all this work and then we're going to push out a final PDF. It's that as we make any single change, we can always click on this report tab and see our report getting made in real time. So I'm just going to sign and submit this. Okay, and then everything is here, and it pulls in my electronic signature, although in the demo model we actually don't have my proper electronic signature, but typically it would have a literal picture of my signature, so you know, just like the standard messy signature that I have. Then it has my name and all my credentials as well. And then this report is a PDF, and so we can print, we can save it. You could open it up in a different software. But this is the physical report. So this is a PDF now of my report, and it has all the information in here. So then I was just going to talk about a couple of cases because, you know, I think talking about the software and talking about the features, it's really easy to not focus on the patients that we're testing. So this is just a couple of examples of kind of how our software, how we use the software in kind of a really productive way. So the first one is just a configured panel. So it's not exciting because we didn't generate a specific panel for this patient. So all that happened is physician saw a kid in the clinic that they suspected had Noonan syndrome. So they went into the EMR and ordered testing for Noonan syndrome. And what that did was it created an order on our end for these 13 genes. And when we upload that worksheet into Genome Ecology, it will automatically say, okay, these are the 13 genes we're going to look at in that patient. We ended up finding a pathogenic alteration that's previously been described, and this is just a mutation in RAF1, so it's a pathogenic mutation. Let me see. Just going to pull these up. So in this case, the variant was previously reported out in dbSNP as pathogenic. And so as I said, it got bucketed in our case into the likely pathogenic call. We went in and changed that to be called pathogenic because it's pretty well documented that this mutation is pathogenic. And then the next time that this mutation comes up in our case, it will be automatically put into this pathogenic tab. And we'll have the ability to link out to this original case that we're looking at right now. But when you look at this, so pull it up in Alamute, you can see that dbSNP calls it pathogenic. Here are all the ClinVar entries. So if you click on all these, you can see that it's always been reported out as pathogenic. Here's the HGMD entry. And if you click on, you could look at the actual papers that are causing it to be called an HGMD. And you know, so from this, when we're going through this case, you know, we felt pretty confident that it was pathogenic. When we look at the pileup, what we see is that, you know, so the calls up to this point are alt, so they're not the reference sequence. And then here, these are all ref. And so this is a heterozygous call. So out of 190 reads, 87 of them were variant. So this is very good. So the variant allele frequency is at 46%, so 47%, which is pretty standard, 50-50 for hereditary disease or for a recessive condition. And so, you know, thankfully we're not looking at somatic alterations here. I know there's probably some people in the audience that are focused on somatic stuff. And so the good thing about inherited diseases are that we're either looking at, you know, 100% or close to it and 50% or close to it. So we don't have to worry about like 5%. There are exceptions to that rule, but in general, it's the case. We sent it off for Sanger confirmation, and it was detected, and so that was filled out in that other module. When we look at our interpretation, we say here that, you know, so this is the interpretation that's going to go into the report. And we could go in and edit this as well. This was done by me on October 5th. And so that's kind of how we would go through this specific variant. And then, you know, in the results summary, there's also a place that you could start working on a results summary here, even though it shows up in the area where it's right next to the report as well. You know, and then just kind of going through these all, usually the genetic counselor does kind of the first pass. And this one, even though it was, let's see, so in this case, you know, it wasn't sent off for Sanger confirmation because it was a benign variant. This is an intronic variant, and so usually to look at intronic effects, we'll pull up all the splice algorithms to see what the significance is. Standard recommendations, and then the report. And you know what we do is we go in and we finalize it. Oops. and then we have an actual report. So this is, so it really shows how easy it is to go through these cases in genome oncology. It takes, it's a team effort. So my view when I'm looking through these is different than the view of the person that's in charge of the technical end. So the technical end, what they're spending their time in is the QC area. And so they'll get their own view, and then the genetic counselor has their own view where they're working on the initial interpretations and kind of the initial workup of the variants, going through all the benign stuff, all the common stuff, all the kind of basic interpretations. And then it gets on to me, and I'll open it up, and this is like my personal work list that's been assigned to me, and I go in and I basically start signing out the cases. So that was the first one. It's very simple. The second case was a personalized sequencing panel by phenotype. So this one, we searched for lactic acidosis, like I showed. As I said, there were 106 genes, so 93 of them are on our panel, 13 are not. After the test, we found that one amplicon had low coverage. And what we did is we identified and reported 3 clinically significant alterations. So there were 2 alterations in a gene called FOXRED1. This is autosomal recessive inheritance. So there's 1 pathogenic mutation, 1 VUS. And then there was a second gene that we found the VUS in. And this is also an autosomal recessive disease. I'm going to go through this just because it kind of highlights one of the features that we were really kind of anxious to get from Genome Ecology, and it's been one of the features that we really like. So in this case, so we have these 3 alterations, and so we can actually change the order. So, as I said, we had a likely pathogenic alteration, a VUS, both in FOXRED1, and then an uncertain finding in SUCLG1. If I want to change this order and I want to bump this one up to the top, I could do that really easily, and then that'll get reflected in the report as well. So there's different situations that you would want to do something like that, and it all comes back to the case of autosomal recessive inheritance. Sometimes having a VUS in a gene that you've found a pathogenic mutation in is more significant than finding out somebody's a carrier in a different gene. And so you would want your report to reflect the significance and the importance of each finding. But this is that case, and so these are all the genes that we looked at, and this was determined from just using that human phenotype ontology for lactic acidosis. And then you see the variants, and we could basically go in and finalize this one if we wanted to. And then, let's see. So this third case is one where, so it was by phenotype also for ectodermal dysplasia. This was based on human phenotype ontology search for ectodermal dysplasia and an OMIM search also for ectodermal dysplasia. Whenever there's an overlap, we're not looking at the same gene twice. We're only going to look at it one time, and it's only going to show up as being one time. But we wanted to make sure we were covering all the genes because OMIM's going to list things different than human phenotype ontology. So there were 19 genes. 1 amplicon had low coverage. We ended up reporting out 3 clinically relevant alterations. So TP63, which is autosomal dominant, there's 1 pathogenic mutation, and then Wnt10A, which is autosomal recessive, reported out 1 pathogenic mutation and 1 VUS. This last case is the one that I was trying to show because I think it really highlights the benefits of taking the array data, but we were running into technical problems early on. So we brought in the areas of homozygosity, which was around 11.1% in this case, and that was determined by chromosomal microarray. Then we used the human phenotype ontology database on those regions of interest for hypotonia. So we said, okay, there's like thousands of genes in this 11% of the genome in this patient that's homozygous. Only show me the genes that are related to hypotonia. And then out of those genes, we said only show me the ones that are associated with autosomal recessive inheritance because there was a cousin that had a similar phenotype. And then what we did is we then cross-referenced those genes from this patient's regions of interest with the cousin's array data that also had regions of interest, and we narrowed it down to 4 genes that were on both patients, that were homozygous on both patients from array. And that was after kind of all this sorting. And this is all done before the test, so 4 genes. So after the test, there were 4 amplicons in those 4 genes that had low coverage. And we ended up finding that the patient was homozygous for this CULQ mutation. The interesting thing in this case is that patients who are diagnosed with these congenital myasthenia syndromes, they usually respond well to acetylcholinesterase inhibitors. But patients with mutations in CULQ, if they take those same medications, then they have all sorts of detrimental issues. So it's bad medication for patients with mutations in this gene. And so treatment was stopped in this patient because of this molecular report, and then the cousin was tested as well. So, this really highlights the benefits of starting with array data, because instead of looking at, and it was a huge number of genes if we looked at all the genes that were associated with hypotonia, or the vice versa situation. If we look at all the genes in the 11.1% homozygosity, we end up with a ton of genes. And so in this case, we were able to get it down to 1 homozygous mutation, which is pretty cool. And it was only looking at 4 genes. So the patient didn't pay for hundreds of genes. The physician didn't get a bunch of uncertain significance findings in hundreds of genes. And we didn't have to look at hundreds of genes. So it's not a trivial amount of work to look at variants in a gene. And so the more genes that you look at, the more work it is for the lab. And so we are always favoring to focus the panels on the patient's clinical phenotype, and then if negative, then expand to a bigger panel. And I'll talk about that in a second, how we actually use it. So this is just kind of how we implemented it. So this was a big head scratcher. We kind of changed the way that testing was done. So before, people would order testing, and the reference labs would set price, and oftentimes the price reflected kind of how much they could get for a given test. So a BRCA1 test, sequencing test, even though it's 1 gene, may actually cost 5 times as much as, whatever CULQ sequencing, just because there's going to be fewer people that are ordering CULQ, and the lab knows that they're not going to be able to get the money that you're going to get from BRCA1 and 2. And so we couldn't really do that because every patient that comes in is going to be a different set of genes. And so what we had to do was figure out a pricing scheme based on the number of genes. And so it doesn't matter what the gene content is. It doesn't matter how big the genes are, although it's a pain to look at really, really big genes. But it's the same price. So you could be looking at Titin, or you could be looking at a single exon gene. You'll pay the same price. And then we basically created this hierarchy of ordering. And so our thinking was that if you're ordering 1 gene, you're really thinking about 1 disease. If you're ordering 2 to 5 genes, you're still probably thinking about 1 disease, just may have several genes associated with. If you're ordering 6 to 15 genes, you're probably in kind of a very narrow phenotype. Or there are some diseases where there's that many genes associated with a single disease state. Once you get above that and you're in the 16 to 50 and greater, then you're really talking about a phenotype panel. And so, this is your epilepsy, this is your workup for cardiomyopathy, this is your workup for, some kind of more broad phenotype. And it goes all the way up to 500 genes. So we do not actually offer a 4,800 gene panel. So we are testing 4,800 genes, but nobody can order 4,800 genes from us. And that's really because there is not a huge desire for anybody to order 4,800 genes. So we get a handful of whole exomes, but the whole exomes that we're sending, and these are, sorry, by we get, I mean they pass through the lab. We don't actually test whole exomes. And so, those whole exomes are used as test of last resort. And in that case, where there's a potential for gene discovery, a 4,800 gene panel is not going to have as much clinical utility as a 25,000 gene panel as a whole exome. It just doesn't make sense to order a medical exome if you don't know what you're looking at, because you want to have the benefits of the exome backbone. And so, so we don't offer 4,800 genes. We only offer up to 500 genes. The benefits of this strategy, though, are that if you jump from one bracket to the other, which we start having that. So we test a patient and they've ordered 15 genes, and then they want us to look at an additional 80 genes. That brings them into this 51 to 100 gene bracket. And so all that they pay on the second look is the difference of jumping from here to here. And so the benefit is that they're not paying any more in that approach than if they just ordered the 51 to 100. And so it really favors smart ordering, where you're only looking at genes that are relevant to your patient. You're not trying to get what's the most comprehensive set of genes I could possibly order at one time. And if you want us to look at, say that somebody orders 51 to 100 genes, and they've actually only told us they want us to look at 60 genes, and then they're negative, and they're like, oh, you know, what about these other 10 genes? You know, we're still in that same 51 to 100 bracket. So they get it for free. So we'll look at it and we'll add it onto the report, and you know, and we'll let them know whether any variants were found the same way as if they ordered it from scratch. When it's by phenotype, we actually only offer it for 16 genes and up. Our thinking is that, you know, as I said, the other cases you're really talking about disease state, and the physician should be able to figure out which gene they want to be looking at. Our physicians are geneticists for the most part, and so we like to think that they all have the ability to come up at least with the genes that are associated with single disease. This is data from about a month ago, maybe 6 weeks ago, and we were right below 300 orders to date. The vast majority, which is this big blue section of the pie, 63% are personalized sequencing panels by gene list. So this means physician actually gave us a list of genes for 63% of them. There's some where it's by phenotype, but it's a small number. And then it's kind of just a big spread of all the 29 configured tests. And so this really reflects the importance of having flexibility in your ordering if you're offering something like this, because we never would have been able to account for these 63%. And that's over, you know, that's the majority of testing wouldn't have been covered by the 29 most common tests. Looking at the number of genes that are being ordered, so this is a reflection really of how long we've been up and running, which is not very long. So we went live in the beginning of February. And so what you're seeing here is people are actually ordering intelligently. So they're starting with single gene targets, like here, 24% are one gene, 26% are 2 to 5 genes. And then they're gradually starting to have us add on more and more. So we're starting to get to the point where, and it takes a while because nothing is really fast in genetics. And so, because in general, things aren't changing so much in genetics. And so if a patient's seen day one and they order 2 to 5 genes, they may 6 months from then come back to genetics, and then they will add on an additional 20 or 30 or 50 genes. And so we're finally at that point where we're starting to see a lot of secondary orders. So we've looked at a set of genes, and now we're looking at additional genes. But at the time of this, you can see that it actually was not the case, where most people are favoring the smaller number. So that's it. So this is our group. So you know, Joe Devaney and Bea Meltzer, so they're the technical people. So you know, so Bea is the one that actually made this whole assay work. Joe's the one that made every piece of the bioinformatics and kind of all these different components work together and kind of had the lead on everything. Mary Beth is a genetic counselor who does like 95% of the work. And Christina is the other director in the lab. 2 of us run the lab together. And this is my contact info if anybody has questions. I'm always open to answer questions very honestly. And thank you all for your time.